Perfect. Okay, I put it here. So again, we'll be talking about Cancogen today, and I think we all know very well by now that we are all facing this challenging storm, but we are not all in the same boat. We see clearly that people are responding differently when they get infected, that symptoms are very different, that there is a lot of variability in how this infection is presented. And basically our goal with Cancogen is to see how science and technology can help us understand and control the COVID pandemic. So Cancogen was funded and created in April 2020 through Genome Canada and was initially awarded $40 million to sequence 10,000 host genomes and 150,000 viral genomes. And the objective is that the data will help us inform policy and public health decisions, and that will also help us advance drug and vaccine development. Now, from those $40 million that were invested in Cancogen, there are $20 million that will go for viral sequencing and $20 million that go for host or human sequencing. The goals, the overall goals of Cancogen are to coordinate the SARS-CoV-2 or viral genome sequence with the host genome sequence. And I will say that that's one of the unique characteristics when we think about genomics and COVID. There are lots of different initiatives around genomics and COVID in the UK, in the US, and in other parts of the world. But this one in particular is quite unique because it's under the same umbrella. We have both initiatives. We have the virus-seq and the host-seq. Now, as I say, we are planning to sequence 150,000 viral genomes and 10,000 host genomes. And of course, it's clear that, you know, the virus for the size of the virus is only 30,000 base pairs. That's why we can do much more sequencing with the $20 million, and we're up to 150,000 genomes. Another important objective for us is that, you know, the sequence is great, and having the sequence from the virus and the host is great, but a very important component also is to be able to link that to the clinical, epidemiological, and basically the metadata associated with those samples. Another important objective for us is data sharing at a national level. And I think we all know that that's one of the challenges we have in Canada, that in particular for this type of health related data that is typically stored and managed at a provincial level. So we are really up to the challenge to see how we can advance data sharing across Canada. Another important objective is to build capacity, and that's really to help us prepare for any future COVID-19 outbreaks or for future pandemics. We believe that this is not the first, this is not the last pandemic, and we have to be fully prepared to face any future pandemics. And as I say on the previous slide, the real goal is, of course, there will be potentially publications. There are already a couple of publications that are coming out of this, or few publications coming out of this, but the main goal is not really a scientific goal in terms of publications. The main goal is to be able to inform policy and public health practices in Canada, and also to provide information that will help advance drug discovery and development and vaccine development. Now, this is the structure we have for Cancogen. So it's managed by Genome Canada. And we have a steering committee, an international steering committee with national and international leaders. We have a host-seq implementation committee. Naveed will provide more details about that. And we have a virus-seq implementation committee. You can see the list of members for each of those implementation committees. And each of those two have working groups that are made of internal members of the implementation committee and external experts in some cases. And some of those are for data sharing, for epidemiological data. So we have also another one that is the ethics and governance working group. So those are really informing and reporting to each of the implementation committees. And we created also a coordination committee that is aiming to exactly do coordination between the two implementation committees, virus-seq and host-seq. Now, the steering committee, as you can see in this slide, is composed of international leaders that are coming from the US, Europe, England, that are working on COVID-19, either from the viral side or human side. And we have also representatives from PHAC, CIHR, and other institutions in Canada. Now, I know this is not about VirusSeq, but I just wanted to give you a very quick overview of what we're doing on VirusSeq. And well, here, this graphic just explain why it's important to sequence the virus. As we know, the virus is mutating very often, and as soon as it's just making new copies of the virus and reproducing and just spreading across different individuals, those copies, those changes in these copies of the virus are helping us trace the virus. We have already, we clearly know already that there are 8 different types of the virus, basically 8 different large mutations that are allowing us to track and just trace how the virus is moving across the world, and is giving us these, distributions, geographical distributions of how the virus is spreading. So the data, lots of the genomic data and sequencing data for the virus is being submitted to a public database that's called GSED. As of November 3rd, there were 175,000 sequences that were submitted. So all this is possible because, we have now the technology to sequence this genome, this virus. One of the challenges, of course, is to make sure that these sequences are of high quality. And that's why in Canada, we have been a little bit slow in submitting. I'll show the numbers, but it's because we want to make sure that we have the best possible quality submitted to these international databases. And as I say, the ultimate goal of that data also at GSED is to help us make policy decisions around that will help us control the pandemic. So here are the numbers we had in September. We'll be updating those now at the end of October, but we had already almost 40,500 sequences completed, and we are now around almost 3,000 sequences that have been submitted to this international database called GSED. So as you see here, we have lots of sequences that have been completed, but we're slowly but surely submitting those to the international database GSED. Now, in terms of host-seq, that's our discussion today. We'll start by saying why it's important to sequence the genome of a host. And we can see in this graph that we have clearly observed that, as we see in the right-hand side of the graph and in the top right corner, that there are these people in red here are people of maybe older than 70 years old with different comorbidities, that most of these people will have a more severe presentation of the disease. But we still in that group can find individuals like this yellow little individual that might have a resistance to the infection or might be asymptomatic. And on the other hand, on the down left corner, we see the individuals are basically unasymptomatic or asymptomatic or uninfected. So people that will have or very mild symptoms or basically get exposed to the virus and don't get infected. And most of those will be younger people with no comorbidities, and most of them will have very mild symptoms. But we have the case here of this individual in that group that even being young, even having no comorbidities, will have an extremely severe disease. So what's happening with those individuals? Is there anything in their genome? And there are actually already a few reports that are indicating there are some genes associated with a more severe phenotype, even in individuals that are younger and otherwise healthy. So here, I just wanted to share this slide on a work that's being done at the World Economic Forum on genomics, precision medicine, and their vision statement. And they highlight 5 gaps or challenges in terms of the implementation. And they basically say that the challenge for implementing genomic and precision medicine and genomics in the clinic is not a technical challenge. And I think we're seeing it with HOSIC. We have the technology, and Navid will explain, we have a really great capacity in Canada for sequencing human genomes. We have the technology, we have the capacity, we have the knowledge, but actually we're facing some of the challenges that are described here in this World Economic Forum white paper. The first and I think the most important one is the challenge of data sharing and data interoperability. So again, we will talk about that, what HOSIC is doing, but that certainly is a challenge for us in Canada. The second challenge is the ethical use of the technology, and that's also a point that we specifically have addressed with HOSIC to make sure we have the right ethical and legal framework. Societal trust and engagement. We're working with Cancogen hard on making sure we inform politicians, we inform our funders, and we inform society in general of the work we're doing, and also the potential genomics has to help us understand and control the pandemic. And the fourth one, I would say, that is closely related to us is access and fair pricing. So make sure that these type of technologies is not only for a selected group of people that are in the main cities, or that are in regions that can afford to pay for the sequencing, but that we can really at least with HOSIC have a good representation of the diversity, and have also promote equity in the access to these technologies inside HOSIC. So I think with that, I will leave it now to Navid to give us a little bit more details on the HOSIC. So Navid, all yours.
Okay, thank you, Carolina. Let me get my slides going, and then if I can get this done. Where are we? Here. Okay, so I hope you can now see my slides. Yeah, okay, good. Right, okay, thank you, Carolina. Should just start by thanking Megan, Namrata, Carolina for putting this together. As Carolina says, so my name is Navid Aziz. I serve as the CEO, CSO at CGEN. Just before I start, I'd like to say that I do believe that this is a critical time for all of us to work together and maximize the impact of the federal government's investment and its importance to genomics in Canada. At CGEN, we are fortunate to provide support to researchers in the fight against COVID-19, and we look forward to working with many of you moving forward. So with that, I think what I'd like to do is very quickly take you through a few slides that will hopefully give you some idea of CGEN's capabilities, but also set the scene up for our discussion today as to and give you confidence into how we will get this project done. So as some of you might know, CGEN is Canada's national platform for genome sequence and analysis. We have 3 nodes, Toronto at Hospital for Sick Children, Montreal at McGill University, and in Vancouver at the Genomic Sciences Center. CGEN's vision is to serve as Canada's engine for genomics-enabled research and discovery. And we do that by producing and bringing access to a critical mass of genomic data, hence our project here, and by building and operating a genomics technology platform that enhances Canada's capacity for genome sequencing and analysis. To give you an idea of our impact and scale at which we work, we have just over 200 staff that help serve around 200 PIs every year. Now remember that these PI laboratories, the 2,000 or so PI laboratories, each lab has multiple PIs and multiple research projects going on at the same time. In 2019, we generated around 1,600 terabytes of data, which for you who are interested, it equates to about 15,000 30x human genome equivalents. So that's a lot of data. We invoiced around $40 million just in the last year in fee for service, servicing PIs across Canada. And we published a bunch of papers and trained a lot of people as well. Between the 3 CGEN nodes, we have a capacity to sequence around 40,000 whole genome, again, 30x whole genome equivalents. That's kind of the currency that we measure medical research genomic sequencing in. So 30x whole genome, we have a capacity of doing 40,000 every year. Remember, in HostSeq, we are tasked to do 10,000 over a period of 2 years. And then we have the compute power to go with that. So that's a very quick snapshot of CGen and the impact, just to give an idea of the scale that we work at. Going to HostSeq now, which is the topic for discussion or information today. As Catalina pointed out, if we're part of the CanCoGen network or a consortium with $1 million earmarked for whole genome sequencing of the host, i.e. the human whole genome sequencing. Catalina touched on this. We have a very strong implementation committee that is running the whole project, but more importantly, you see that we have subcommittees listed here. So these subcommittees are formed as the need arises. And as you can see, right now we have 3 very important aspects of the project covered by these 3 subcommittees. We have starting at the bottom, we have the HostSeq data genetic epidemiology subcommittee, is chaired by Lisa Strug out of Toronto, and they are the people who are advising and looking at how we're going to analyze the data set for insights into the genetic effect of or genetic involvement of response to COVID-19. Then we have another very important committee, which is HostSeq Data Storage and Sharing Subcommittee, which is chaired by Stephen Jones in Vancouver. And again, that's the committee that's advising on how we will store and share the data. Stuart will talk a little bit about our data governance a little bit later on. And then most relevant to today's discussion is our study recruitment subcommittee, which is chaired by my colleague Stuart Turvey, who's here, and I'll hand it over to Stuart in a second here. But again, as you see, is a fairly powerful committee representing clinicians and others across Canada who are helping and advising on how we need to recruit studies. One point to remind everybody here is that the design of HostSeq is such, and Stuart will provide more detail, but the design is such that we're recruiting different studies into the program, not individual patients by and large. That's the plan. So before I hand it over to Stuart, just very quickly, the goals of CGen project include whole genome sequencing of 10,000 Canadians to help understand the genomic architecture of the host response to COVID-19. Of course, with the metadata linked to and the clinical information linked and provided by building a national COVID-19 related genomic sequence data banks that will support researchers across Canada. So whether you have samples included or provided for the 10,000 data bank or not, as a Canadian scientist, you will be able to get access to the data bank as it's established. And Stuart will talk about that a bit later. Second being to identify genetic variation that might impact susceptibility and disease severity of COVID-19. Catalina touched upon that, why that is important. And third, also to provide genetic evidence to support therapeutics and vaccine development. So any vaccine and therapeutics that are developed are not only targeted to the right areas to the right genetics, but also to the right population. And with the diversity of the Canadian population, it's very important to have insights of differences in these population, and any therapeutics that target specific areas of a human genome need to be considered as to how is that area of the genome different within different ethnic cities or diversity. So it's important to take that into consideration as well. So quite a important aspect of the analysis. Having said that, I think I'm going to hand it over to Stuart to quickly introduce himself and then take it over with some details on what we've been up to and the study design of the project. Stuart?
Great, thank you, Naveed. So my name is Stuart Turvey. I'm a pediatric immunologist. I work at BC Children's Hospital, and in my everyday job, we harness the power of genomics to diagnose children with immune system problems. So this is sort of a logical extension for me in terms of being able to participate in this important initiative. And I'm delighted to participate and really excited to share the initiative with you and make sure that we have as much awareness of this opportunity as is possible. So I'm sitting talking to you from my basement here in Vancouver, and it sort of feels like I've been here forever. But we haven't really, and I think it's helpful to think how quickly this has come together since from the Prime Minister's announcement in April to setting up the program, being ready to receive and sequence samples in the early summer is really been fantastic, and I think a testament to national will to work together to tackle the COVID-19 pandemic. And so I'm going to take you through what it means to be involved in this, and I think hopefully to reassure you that it's relatively easy and that we're here to partner broadly. So the next slide is from our website, and we'll provide the links there in the chat for this, where we lay out step by step what people need to do to be involved. It's relatively straightforward, and importantly, we're really looking for partnership. This isn't like I don't know, submitting a journal where they just reject you, submitting to a journal or submitting to a grant, which is how my life mostly goes. This is about partnering and seeing how we can work together, perhaps to modify the consents of the project to be eligible for HostSeek. The next slide shows it in a diagrammatic form, which I think is a little easier to understand. So, as Naveed said, HostSeek has been set up to partner with other research projects. So rather than linking directly to research participants who've had the infection, we've taken the strategy to partner with other research programs. This was deliberate, intentional strategy because we knew that, for example, CHR was funding COVID-19 research, provinces were setting up biobanks, and many other initiatives. So we've been actively partnering with those. On the right-hand side in the purple box is really what the HostSeek project is funded to do. It's funded to receive a blood sample from someone who's provided appropriate informed consent to perform the whole genome sequencing, returning the data to that referring investigator and their team, but also depositing that sequence data and the associated clinical metadata into a large national database. And why that's important is that many investigators may not be on the ground recruiting subjects, but may be excellent in bioinformatics and really interested in accessing the database. And so we haven't had our first data release yet, but that's planned. But when those data are available, there'll be a relatively low bar controlled data access process where investigators, industry, and others can access this data to empower a national and international fight against the virus. So really there are multiple ways people can be involved. Right now we're focusing a lot on the recruitment side, but there'll be fantastic opportunities on the data analysis side. On the left-hand side of this figure, we really see what happens. So let's go back. Yeah, perfect. If for people who are running projects, we'll work with them to ensure that their ethics covers the core elements necessary for participation in HostSeek. And on the website, we have a ready-to-go consent form, but also identification of the core elements. And what people can do is actually give us their consent forms. We work with Barton Knoppers at McGill, who is a international expert in ethics and genomics. And we'll give advice on how to modify those consent forms as necessary, and we'll help people achieve the necessary consent. Agreements have to be executed between the investigator and CGEN to allow the sequencing to happen, but it's truly a facilitated and mentored process. The next slide, please. Our eligibility criteria are straightforward. We want people to have evidence of SARS-CoV-2 infection. And for us, that would had initially started as being a positive molecular test from nasal brush. But as technology has evolved and the diagnostic capacity has changed, we'll also be able to enroll people who have positive serology. Individuals need to be able to provide consent, or their legal guardian provide consent. And for teenagers, we will also be needing them to provide their own assent in addition to their parental guardian consent. So exclusion criteria would be no evidence of the infection, relatively straightforward, inability for the individual to provide the informed consent. And then we also don't want to be enrolling people twice. And so we're tracking provincial health numbers, and we'll be able to avoid double enrolling subjects. But again, relatively straightforward inclusion and exclusion criteria. Next slide, please. So we really are open to all. We're actively engaged with about 55 investigators or research programs across the country. Maybe not surprisingly, the numbers are bigger in Ontario and Quebec, where the pandemic first really took hold, but we are actively engaging across the country, and that's the reason for these discussions. And I guess the challenge to everyone on the network, on the call, would be to reach out to us if you have a project, but also use your networks to help people become aware of this opportunity. The next slide. When we look at the people who we've started to work through and to onboard into the project, we see that there's nice diversity in terms of their recruitment setting for those projects, and that the study goals are quite diverse. Next slide. And these are the numbers that we're looking at. So our goal is to recruit 10,000, to sequence 10,000 Canadians with COVID-19 within the next 18 months or so. And the good news is that we're starting to get closer to these numbers with almost 8,000 possible people to be enrolled based on our onboarding strategies to date. The reason we put this number out there is I think, unfortunately, the pandemic is still very active in Canada, and it's likely that we will fill this 10,000 number. So we are wanting people to reach out to us sooner rather than later, so that we're not fully subscribed. The next slide. And this is the numbers of where we are today. I showed you that across the country, we've been enrolling about 55 investigators. 16 of them are actively onboarding with us. That means working through agreements, having the consent forms approved. 3 agreements have been executed to date, and from those studies, 672 samples received and over 500 samples sequenced. So the system's absolutely working, and we will begin to accelerate that as people move through the onboarding to having the executed agreements. And the last slide, I think, is something that's been really important to us, is around this data strategy. This has clearly been identified through white papers and others as a key aspect to ensuring that genomic medicine reaches its full potential. And Dr. Steve Jones leads our data storage and security subcommittee, and have been working very hard to make sure that our governance meets international standards, that we have long-term hosting strategies in place so that this database will last well beyond this pandemic, which will end, and well beyond, and will be able to serve the Canadian community. And I think we're also with this project looking to push and achieve some of the goals that have been articulated, but have been sometimes hard to actually achieve. And one of those has been really pushing hard on a plan for national data linkage, where we'll be able to use the consent of the subjects and their health identifying numbers to be able to link to national administrative data, which will be incredibly powerful for this study, but sets the scene to empower genomic work in Canada in the future. And so with that, I'll just end by saying, you know, if you're an investigator and you're recruiting COVID patients, please talk to us. What you'll get from that will be free whole genome sequencing, which is a remarkable biomarker, and you'll be contributing to a really important Canadian initiative. So we're open for business and delighted to partner with you. Thank you.
Thank you. Thank you, Stuart. I'll just wrap up by saying that we believe that this is an exciting time for genomics in Canada. And as a collective, I think we have a great opportunity to showcase the power and promise of genomics and its role in impacting the everyday lives of all Canadians. So we need your help. As Stuart said, please join us. Thank you very much.
Well, yeah, thank you.